lot about the relationship between ecology and genetics in terms of resilience and restoring the bay, but not so much into the nuts and bolts of restoration practice. So I'm sorry if I misled you. And this research is highly collaborative, and it's been going on for a number of different years. It's led by Dr. Katja Engelhardt up at the Appalachian Lab, also part of UMSES. And then we've had 2 PhD students, Mike Lloyd and Brittany West, working on the overall project, as well as a number of undergraduates and research technicians, and Haley Toomas, Robert Burnett, Paul Widmeyer, and Leslie Peterson. So have all contributed through both helping us with the main research focus and then doing their own independent research projects. So I'll be talking about some of the work that we've done today, but I can't cover it all. And in putting it all together, I feel like we've accomplished a lot and that we've just barely started getting some understanding. So for those of you who are not seagrass experts, seagrass is a generic term that's applied to a lot of the submersed aquatic vegetation. So these are plants that are rooted in the bottom of the bay or stream, wherever they're growing, but remain under the water. They do not emerge from it. None of them are actually grasses, but they're just generically called seagrasses, and they belong to a number of different families, and so they're not necessarily all related, but people generally lump them into things they call seagrasses. And they are important in ecological functioning in aquatic systems in that they provide important nursery habitat. They provide waterfowl food, and in fact the species we work on is in the genus Vallisneria, and the scientific name of canvasback duck is Athaya Vallisneria, indicating the importance of this in its I think in its life history. And then the submersed aquatics also provide the foundation of aquatic food webs. They're important in sediment trapping, nutrient absorption, and shore protection. And so although I just like the plants themselves because they're really cool, they actually do provide really important ecological functions and ecosystem services that benefit humans, that people are really interested in putting economic value on in terms of their help in cleaning up the bay and ameliorating the effects of pollution and sedimentation. So seagrasses, as with many other organisms in the bay, used to be much more abundant than they are now. And Orth and Moore in 1983 published this a map similar to this. We modified it a little bit to make it a little bit prettier than it was in 1983, where all the dark blue areas were extensive seagrass beds, but they have primarily they have majorly declined in these areas such that we only have a few remnant patches left. And the areas in lavender were the only areas that were relatively unaffected. And nothing is unaffected, but just relatively so. And most of this decline is due to 2 culprits. One is sedimentation, and the other is nutrient. The text didn't show it up. Is nutrient loading, and both of these have the effect of cutting sunlight that penetrates through the water, and then the seagrasses cannot photosynthesize enough to stay alive. So both the sedimentation blocking the light, and then the eutrophication from supplemental nutrients causes algal blooms that block light, and then also cause epiphytes to grow on the seagrasses, and so their photosynthesis is cut to the point that they can't survive. And a lot of these 2 effects, or these 2 triggers, come from conversion of the 64,000 square mile watershed from a state that looked like this to a state that looks more like this, or this, or this. And so the runoff that's coming from agriculture and urban areas is degrading the water quality that leads to the bay, and then the water quality of the bay itself. So by the 1970s, the levels of seagrass abundance or submersed aquatic vegetation abundance were known to be extremely low in terms of the total number of hectares that were detected. And it was about that time that the Virginia Institute of Marine Science started a monitoring program where they've flown aerial photography of the Chesapeake Bay to document the extent of seagrasses, or submersed aquatics in general. So through time, through some pollution reduction and sedimentation reduction, the amount of seagrass has increased over time. But in the late 80s, early 90s, it sort of plateaued and it's bounced around at this level for quite a long time now. And so one of the questions that a lot of people have had is, why has it hit this plateau? Is it simply that water quality is still not to the point that the vegetation can expand and persist better, or is there something else going on? And to give you some perspective, the levels of seagrass abundance are still far beyond the restoration goal that was set, where this was supposed to be achieved by 2010. The goal was to get up to about 80,000 hectares of submersed aquatics in the bay. We're far below that, and we're even farther below the historic SAV coverage, which is estimated to be about here. So we're still greatly impaired in terms of the seagrass abundance in the bay. And then if we look at the way that the Department of Natural Resources quantifies things, they look at the watersheds that are leading to the bay, and they rate each of the watersheds in terms of, here it's submersed aquatic vegetation habitat, which is based on water clarity, dissolved inorganic nitrogen, phosphorus, chlorophyll A, and suspended solids. And most of the watersheds receive a low score, so this is better going down, so the lighter colors are better. So most of the watersheds are highly impaired, and these are old data, but these are all that are available on the web anyway. And then if we look at it in terms of submersed aquatic vegetation abundance, and we look at the percentage of the goal that's been achieved for each watershed, which is a function of the watershed, the suitable habitat in that watershed in terms of water depth, sediment type, wave exposure, and then how much SAV was there historically. Most of the watersheds are at about 10% of their watershed goal for the amount of SAV that's present in each of the sub-watersheds. So, and all of this is despite extensive efforts to clean up the bay and to directly restore SAV in the bay. And really heroic efforts. Unfortunately, most of the direct restoration efforts are relatively small compared to the magnitude of the issue, and they're very intensive in terms of labor and cost to get this done. So in 2010, the sort of lack of other political will to clean up the bay led to establishment of the total maximum daily load for the Chesapeake Bay. And this is established by the Environmental Protection Agency, but involves working with multiple states, all the states in the watershed and the District of Columbia, and local partners to have a way to have more accountability in cleaning up the bay and actually move to reducing sediment and reducing nutrient input into the bay. And it's the so-called, you've probably heard about it, it's the so-called pollution diet for the bay. So the idea here is that rather than doing direct restoration efforts, if we clean up the water, the rest will follow. Unfortunately, since that time, even it's not been in place for all that long, but since that time, the health of the bay has not increased that much. So the most recent Chesapeake Bay Foundation report says we have an improvement in the bay. We've gone up 1 point on a 100-point scale from a score of 32, and my students who get a 32 out of 100 are not all that happy. But we've gone up 1 point in their overall health index. And if we look at all of the things that go into that health index, there are pollution categories, habitat categories, and fisheries. we're not doing very well in most of those categories. We're either the same or maybe a little bit better, but we're still, if you're going up from an F to a D in your report card, it's not really all that promising. And then the only one that has declined over the last 2 years in their report are the seagrasses. And a lot of this was thought to be due to Hurricane Lee, and some of the, there are some dead zones that have cropped up that have caused declines in Zostra over that time frame. But so all of these indicators are mostly the same, or a little bit better, with the seagrasses actually declining over the last couple of years, unfortunately. So it may be that if we simply clean up the bay and we reduce the sediment, we reduce the eutrophication, that the photosynthesis capabilities of these plants will come back and they will recover. There's also a possibility that because of the dramatic declines we've seen, that there is some compromise, that these species are actually compromised in terms of their ability to acclimate to or tolerate these conditions, or to adapt in the future. And this is related to potential loss of genetic diversity that would have been present in the bay prior to these declines. So if we think of what it takes for a species to survive in either compromised conditions or novel conditions that the species has not experienced previously, the species can either tolerate the conditions or acclimate to those conditions. And that's with the current phenotypes that are present, or the current individuals that are present. Alternatively, you can adapt through natural selection, where you have some individuals that may be tolerant of conditions, and then those individuals leave more offspring, and so you have an actual change in the composition of your species through time in response to these changed conditions. Some species can also move to new locations. Animals can do that a little bit better than plants can, but plants can do it through seed dispersal. You can become locally extirpated and still exist outside of the area, or ultimately extinction is the final option if you can't do any of the, if you can't tolerate, adapt, or move. So it's this tolerate and acclimation that we're interested in. Tolerating and acclimating to conditions, and then adapting to conditions that we're interested in terms of conferring resilience or persistence to novel conditions. And so our options, there are multiple options for this resilience. So if we look at, on the x-axis here, we look at some niche axis or environmental gradient. And so we're looking at a tolerance of an individual to some sort of environmental variation. We can have single individuals that have very broad tolerances. So an individual might have a high tolerance of shading, high tolerance of salinity, high tolerance of any sort of environmental factor that might be difficult for other individuals. Alternatively, we could have individuals having very narrow tolerances, but variation among individuals in those tolerances. So this population, through different individuals, has as broad a tolerance to environmental variation as the top, but it's because you have multiple individuals. Each of those may not be able to acclimate very well, but as a whole, that population has individuals that would survive under a range of conditions. Our third option is that we have low variation among individuals and within individuals. And so you would have very low potential for acclimation or adaptation under the third scenario. And so what we're interested in is the diversity within populations, both genetically and phenotypically, to understand whether we have a situation like this where we have many individuals but they all have narrow tolerances, or we have multiple genotypes or multiple genetic individuals, but narrow tolerances, okay? So the genetic diversity is important for resilience and restoration from a number of perspectives. So we're interested in levels of inbreeding within individuals because this is often correlated with that breadth of tolerance of environmental variation. If you have highly homozygous individuals, they often have very narrow specificity. So we're interested in the number of alleles or genotypes in populations, and there's lots of evidence from the Zostera world, the eelgrass world, where low numbers of genotypes or alleles decrease growth and survival. And then if you don't have very many different genotypes, you don't have much opportunity for acclimation or adaptation to different conditions unless you can acclimate by having a broad tolerance, but you have no potential for adaptation. And then we're also interested in local adaptation of individuals where we have differentiation across populations, and then potential for species that, or individuals that are suitable in one situation may not have high fitness in another environment. And so they're highly adapted to where they are growing, but may not do very well if they were planted elsewhere. So we're going to look at these three issues in our favorite species, which is Vallisneria americana. So this is one of the meadow-forming seagrasses. So where you find it, it can often form very extensive and thick beds where that could be the only species there for quite a large area. It can also grow intermixed with other species, but it's one of the kind of foundational species that tends to persist from year to year more than some of the other species, which you can have a lot more boom and bust. It's not completely persistent, but it's more so than some of the other species. So male and female flowers occur on different individuals. So it takes at least 2 different clones to have sexual reproduction. The female flowers float on the surface when they're in bloom. And you can see lots of female flowers floating here. The male flowers are at the base of the plant, and then they release the pollen to the surface of the water, and it floats sometimes sometimes just open like this, and sometimes in a little cup-like thing. But generally, the pollen you'll see floating on the surface, and it has to meet one of the female flowers in order for pollination to occur. So it's kind of an interesting pollination syndrome. They also reproduce vegetatively, so you'll have one ramette here that can produce multiple, it has underground stems and then can produce multiple other ramettes, so these are all genetically identical, and then at the end of the season they'll produce these little overwintering structures called turions. A lot of people also call them tubers, but they're not technically tubers. So they produce little overwintering structures so that, and then in the following year, new ramettes will grow up. And so you can get these extensive areas that are actually all one clone that are spread by rhizomes, which is how it can come to dominate large areas.
So is the sexual reproduction sort of random? Do they meet, or are they close to each other?
Well, right. So it's as with most plants, things that are closer to each other are more like a female plant closer to a male is more likely to be sired by that male than something that's far away. So there's definitely spatial structure, and we've done. Well, the pollen from a male plant has to float on the water and then intersect one of the female plants. So it's pretty dicey. And we've done some, I wasn't going to talk about it today, but we've done some paternity analysis where we've looked at how far away from the female flower the pollen comes, and it's different in different populations, but it's generally less than about 20 meters. So, and it can be much less than that, and it depends on whether it's a river, open bay, or a protected bay, sort of how much water movement you have, how far the pollen flows, but it's generally not very far. Okay, so our general approach has been to start by collecting leaf tissue and then ramets with the roots from the field. We've then genotyped all of our collections so we know their genetic identity and we can get down to individuals. And then we use those ramets that we've collected, we grow them up in the greenhouse. So we're growing everything we've genotyped, or everything we have growing in the greenhouse, we know its genetic identity. And then we conduct experiments on those genotyped individuals to link the genetic diversity to ecological characteristics that are important for persistence and in the bay. And these are the little overwintering structures that allow us to, so we grow the plants up, let them die back in the winter, and then we harvest those turions, and then that gives us genetically identical individuals that we can use in well-replicated experiments and put the same genetic individuals in different environmental situations. So it's a wonderful plant to work with. I've mostly in my career worked with endangered species, and you can't get them to germinate, you can't get permits to collect that many individuals. There's just it's not that experimentally tractable, and this is a fabulous, fabulous plant. So, and then what I mentioned that we have done that I wasn't going to talk about today was we've also collected fruits and then genotyped those seeds and used a method called 2-gener, which allows you to look at how the spatial distribution of potential pollen, the pollen pool that sired these fruits. But that was too much to put into this. So what I'm going to do is talk about the genetic information first, and then some of the experiments we've done to look at the relationship of the genotypic diversity we see back to ecological factors that matter for persistence of Vallisneria in the bay. So from our initial collecting, the royal we, this is a lot of Mike Lloyd's work. We collected 675 samples from 26 sites throughout the bay. And most of these sites are tidal, but the Potomac, we also sampled from the Potomac above the fall line to get some information on that freshwater reach of the Potomac. And so I'm going to go through the several different aspects of genetic diversity. I won't bog you down with all the detail, but I'll show some of the highlights. So as I said, the first thing we were interested in is how many genotypes are there in a population. And so that tells us the number of genetically unique individuals that are present at any different sampling location. And what we find is that generally in the main part of the bay, we have high genotypic diversity, and it can range generally from about 70% to all of the individuals being genetically unique out of our 30 samples. But when we get up into the Potomac above the fall line, we find that many of those sites are extremely depauperate in terms of the genotypic diversity. So these two populations here, or these two sites, this one has 2 different genotypes and this one has 3 out of 30 individuals. This one, they're all identical. So very low genotypic diversity. So if you went out, you might see a large, and these are not small populations necessarily. The Potomac looks like one, mostly continuous Vallisneria bed. You can see thousands of ramets, and you would think, wow, this is a great, great population. But in fact, in this case, almost everything up here is genetically identical. And so that has a lot of implications for future change. So one of our questions that I'll get to at the end is, are these really dominant genotypes, are they the ones with really broad tolerances, or are they just the only ones that are left after a huge decline, and that they're basically the consequence of a genetic bottleneck, and they just happen to be doing fine now, but may not be in the future, and we're trying to get at that experimentally. So we have a lot of variation among sites in terms of the amount of genotypic diversity there. Yeah?
How
It lasts we don't know in the field, but in the lab, it's about 24 hours. So it doesn't last very long in the greenhouse. Well, and so the genotypic diversity is, so we're not, basically it's the same exact clone. There's no, and they're one sex, so either they're male or female. So there is no sexual reproduction happening where you have one clone. And I haven't looked actually to see whether these ones that have, 2 and 3 clones, whether they're all one sex or not. We now know for most of our clones which sex they are because we've been growing them up in the greenhouse and they've flowered. So,
sort of down
so down here, so this is below the fall line. So there is obviously a difference between one-way water flow and tidal influence here, right? Yeah. So, but it's not entirely. I mean, there's all, well, we'll get to it with heterozygosity, which is getting at how close are the things that are mating. So this is just the number of totally different genotypes.
What is the generation time for that?
Generation time, no idea. No idea, because they're clonal. So an individual can persist, and so if you take it's not clear what the actual generation time is, because they can. So but they will flower the first year. They can flower the first year that they germinate, and there seems to be a minimum in the greenhouse. They get to a minimum size, and then they'll flower in 1 year. But then they can persist, and it's. So let me show you one other thing so we have huge variation in genotypic diversity within populations. Also, the genotypic diversity, at least in the Potomac, is shared extensively among sites. So the lines here show the connections. So if there's a line between one dot and another, it means they share genotypes, and they go directly across land because it's just easier to draw that way with the computer, but they're obviously not traveling across land. So so these sites are sharing genotypes. So not only can one site be dominated by 1 or 2 genotypes, but this entire area is dominated by a few. So we have 4 genotypes that occur across multiple sites. Yes, Dave?
And so those are the only ones that share? All the other ones that you don't have connections don't share anything? Is that why they don't have lines, or did you just do those?
I'm pretty sure they're not sharing, and I need to double check that, but they do not share extensively. It's fundamentally different in the Potomac versus the whole rest of the bay. Yeah. And we have not done the Susquehanna, which I really would like to get into for a number of different reasons, but I really want to know whether this is a river phenomenon but it's fundamentally different in the bay. They do not share, they have many more genotypes within populations, and they do not share substantially among, or I should say sites. So if we look at this genotype creatively named genotype number 1, its frequency and this population, it's half of the individuals we sampled were that one genotype. So you go down to the next population, 80% of so basically 1 of 2 was, how it's 53% then I don't know, was one genotype, 80% is another. And then as we go down, we see almost 50% of the Hancock population is that genotype, drops way down here at Williamsport, and then comes back up here at Brunswick, and then is also present at Point of Rocks. So this one genotype spans this entire area. So when you ask about generation time, who knows? I mean, this thing, who knows how long that one genotype has been present in this river system.
Just for context, seagrasses that ocean, in the Mediterranean, they've estimated that clone, 200,000 years. There are single clones that are thousands of kilometers.
Right.
Long. So
Right. So they just,
this isn't,
it's
right.
yeah.
This isn't unique. This is the biology of these
Yeah.
unique plants.
So
this time scale of adaptation is not necessarily different than the time scale of the acclimation.
it's very different. Yeah. Yeah. Well, in any system, I mean, acclimating you can hopefully do on a 1 to 2 to 3 year, you're acclimating in real time, otherwise you would die. Adaptation is going to take, it's going to take a while. So if you can't acclimate, you can't adapt. So.
But it seems like you could, if something is living for thousands of years, but it also has some sexual reproduction on the side, you could have the reverse, where it's long, it's not right?
Right, where it basically, if it's persisted through long periods of time, it is tolerant of a wide range of conditions that it can hang out there. Is that what you're saying?
Well, I'm saying actually that you could have like adaptation, because genetically it might be a random mating or sexual reproduction that's occurring, and that could be you could have adaptation faster than you could have, because this whole thing is living for thousands of years.
And you're saying adaptation would be faster than?
It just seems like it's interesting that the scales aren't really clear.
Because an individual occurrence in one spot might have the genetic mating and then it could die off, but so it's passed on.
Right, and the individual is maybe living for thousands of years, but it's
Right.
Yeah.
Right.
You usually think of it the reverse. The adaptation is taking a long time, and then the acclimation is really short term.
Good point.
So I'm not catching it. So you're saying
that if you have this clone that can live for thousands of
yes.
years, then its lifespan is longer than the process at which genetic.
At which adaptation? Right. And so you could have. So then are these very extensive clones the ones that are more? They can acclimate better, and then you have all this, experimentation going along through the sexual reproduction on the side. Some of those persist, some of those don't. Is that sort of?
Yeah.
Yeah. No, it's really.
The timescales are not clear.
Right.
Yeah.
Right. Yeah. Yep. So then we have this second clone here that is shared by these populations and is the clone that makes up all of the Pennyfield lock site. So we have a lot of variation site to site, and in terms of the dominance of any particular clone, okay, which does have implications for potential for sexual reproduction, adaptation versus acclimation. So,
how do you, if they're really thousands of years old, could you have adaptation within a clone?
You can have mutation. You can have mutation within a clone,
and then all of a sudden it would look different.
Yes, and you can get somatic mutations, whether those confer adaptation or whether or not. Generally, most random mutations are going to be deleterious, but you definitely have mutations over those time scales. So, yeah. And then the other thing I should point out that often when you think of clonal growth or you think of a clone extending over a particular area, you think of it growing out incrementally. And it's probably the case here where you have ramets that have broken off during flooding events, and then those get carried downstream and they can reroot. They can get deposited and reroot further down. So it's not like this is all necessarily one continuous clone. Okay? So this is just the genotypic diversity slide once again, because I'm going to move on to allelic diversity, and I wanted to have this to flip back and forth too. So if we look at the allelic diversity now within populations, of course this is going to be a function of how many genotypes you have there. If you have only one genotype, you can only have a maximum of 2 alleles per locus, right? So you're necessarily limited. But some of the interesting things, there's some interesting contrasts here where here we have sites that have a fair degree of genotypic diversity, but very little allelic diversity, meaning the genotypes that are there are very similar to each other and share a lot of alleles. So although this looks better, looks more diverse in terms of number of genotypes, they're all actually fairly similar to each other. And we see that in a couple of instances where we have fairly high, sorry, went the wrong way. We have fairly high genotypic diversity and then lower allelic diversity. But in general, we see the same thing as before, that these bay populations have higher diversity, the Potomac are the lowest, and then these tidal Potomac are intermediate. Then when we look at observed heterozygosity, which is often, it's often a measure of, well, homozygosity tends to be bad because you tend to have more deleterious alleles. If they're recessive, they can be expressed. You have 2 copies of the same deleterious allele. And so we like to see a fair amount of heterozygosity. And so we see in the Potomac actually fairly low heterozygosity. Again, the bay populations seem to be a lot more genetically diverse, but the Potomac populations seem to be struggling with heterozygosity. Now, there's not all heterozygosity. Actually, let me say one more thing. So, the only places where heterozygosity is significantly less than expected are these 3 populations. The other ones, the heterozygosity is what you would expect based on the allele frequencies in the population. So the only ones that show a deficit are these 3 in the lower Potomac. Now there's 2 ways to get that. One is through inbreeding, and the other is through mixing 2 distinct populations, 2 or more distinct populations, where you have individuals coming together that are having different origins, and then because they have different alleles, what your expectation is, you have a certain, based on Hardy-Weinberg, that you'll have a certain degree of heterozygosity. But if those individuals have come from 2 different places and they have not mated yet, and so you don't have their offspring, you'll have what's called a Walland effect, where you have a lower heterozygosity than you expect based on your allele frequencies. That has actually nothing to do with inbreeding. And this is potentially what's going on in this zone, and I'll show you why that may be in just a bit. Okay. And only one population showed signs of a bottleneck, which would be recent decline in numbers of individuals that leads to lowered allelic diversity. So all of the others seem to be relatively fine in terms of what we would want to see in terms of genetics. And so when we started this, there was no information on the genetic diversity of the species in the bay. And it really could have ranged from, it was so depauperate due to the past declines that you basically had 1 clone throughout the bay, or very little genetic diversity, or that every site was completely different. We really had no idea what to expect. And I would have leaned towards the very little differences, very few differences across the bay, just given the hydrologic mixing and everything. But so we didn't really know what to expect when we started. So.
It's hard for me to think about bottlenecks.
Okay.
There's, some of these populations are just 1 clone. I mean, I think of a bottleneck as a population going down to just a few individuals. So it seems like if there's just
if there's one individual, 1 clone is 1 individual.
Right, but if they're all, okay. So
right, genetically,
right.
It's 1 individual.
So that's why it seems like you should have more bottlenecks since some of these populations.
But but the way the test works, it's it basically you have a recent decline, and you have, and I'm not remembering what the actual test is, Dave.
It's mostly about loss of alleles.
It is about loss of alleles, but I can't remember what the ratio is. I'm blanking on it, but it's whether you have.
Well,
yeah. I'm sorry, that's embarrassing. I'm totally blanking on it.
No, it's
And that's
okay.
going to be on the web. Woohoo!
We can flip that.
So, okay. So the next thing we wanted to look at was the potential for local adaptation. And what I'm showing you now does not demonstrate local adaptation, but it's one of the first requirements, which is, do you have genetic differences across space within your within the bay? And so these are the results from a program two programs, one called Structurama, one called Structure. And what we're looking at here is each of the black boxes with a label underneath is one of our sampling sites. So our sampling sites are arrayed across the x-axis here. And then the colors, it's easier to see over here. Each of the boxes is made up of a bunch of different lines that all represent one of the genotypes in the population. So our sampling effort was actually equal across all the populations. The boxes are narrower or broader based on the number of genotypes in those populations. And the colors of each of these lines refers to the probability that that individual came from a particular quote, genetic population. So the first thing the program does is it takes all the data, says how many populations how many populations did these individuals come from? And in this case, the program said that 3 was the most likely number, and that we had a group that are indicated by blue, a group that are indicated by red, and then this group here. And these were fairly similar to each other, but there was a secondary group where these two were split apart. And you can see the similarity between them by the number of green, the amount of green in some of these populations, and indicating a lot of similarity between these two. So we're saying there are 3 major genetic regions, and then one of those is split into 2. If we look at how that arrays in space, the blue are the Northern Bay populations, the red are the Central Bay, yellow is Lower Potomac, and green is Upper Potomac. So and so you can see the similarities between the Upper and Lower Potomac, but also that they are different from each other. And that you also have some more influence of the the Central Bay and the Northern Bay in these populations, which is why I was saying it was potential that we had this Walland effect where we've got these different sources for these Lower Potomac populations, that we have Upper Potomac influence, then we also have influence from other parts of the bay, and so we may just not be in Hardy-Weinberg equilibrium yet. And that may be the cause of that heterozygote deficit there. Okay. So these are based on the microsatellite data, but gave us some strong indications that we do have some strong genetic differences in the bay. Okay. So summarized to this point, overall our genetic diversity levels are above levels that would be of great concern. So when I'm working with species that are endangered and occur in very, very small populations and are likely to be genetically depauperate, we're above those levels. So that's really good news that we have in the bay as a whole a lot of genetic diversity to work with. Particular sites, though, have relatively low genetic diversity, and it's especially those Potomac River sites. Only one site had that bottleneck evident, but that we do have evidence for potential for local adaptation given that we have genetic differences. So now I'm going to move to some of our trying to link this genetic information to ecological performance. And we do that through a series of very sophisticated and high-tech experiments. And basically, we work with a lot of mud. And again, we take advantage of these turions that the plant produces to be able to do these very nice replicated experiments. And we grow the plants in the greenhouse or in growth chambers and under different conditions. So I'll explain a couple of the experiments that we've done. So in the first set of experiments, and this one was really done by Katja Engelhardt up at the Appalachian Lab. And so she took all of the clones that we had genotyped, and then we selected a certain subset of individuals, and we focused on 2 populations that we had a lot of different turions for, so we could have enough individuals to do these experiments. We did include other populations, but we focused on these 2. And so there's one Central Bay population, Hawks Cove, and then one Potomac population, Piscataway Park. So we tried to do multiple things at the same time. We first looked at the effect of individual heterozygosity on growth and reproduction, because this has been shown in numerous species to be important. So because we genotyped the individuals, we could select low heterozygosity individuals and high individuals that were heterozygous at more loci. And then we took 49 genotypes from 14 sites. But again, most of those were from either Hawks Cove or Piscataway. We also looked at the effect of genotypic diversity by planting 2 genotypes together, multiple combinations of 2 genotypes, multiple combinations of 4 genotypes, and multiple combinations of 8 genotypes together to look at a genotypic diversity effect. And then we looked at local adaptation based on performance in home versus away soil. So we looked at we looked at Hawks Cove and Piscataway clones in Piscataway soil and in Hawks Cove soil. And so these were individual single clones planted in each experiment, or each mesocosm. These were different numbers of individuals in each one. We did 516 monoculture mesocosms and 120 multi-genotype mesocosms. And so I'll just run through the basic results, because there are so many. So we looked at whether heterozygosity, genotypic identity, or population source in the monoculture experiments affected leaf length, number of ramets, the rhizome length, tuber biomass, or the length and frequency of flowering. So we looked at all of these things for each of these characteristics. We find equivocal evidence for effect of heterozygosity on leaf length and tuber biomass, but generally heterozygosity was not that important in explaining any of these characteristics. Genotypic identity was extremely important, as was population source. And so what we're seeing in our experiments kind of over and over is there are some really studly clones and there are some real wimpy ones. And then often that, not always, but often that relates back to the population from which they came, but it's not a function of heterozygosity of those individuals. And this is with the sample sizes I've shown you, so it's, it's limited, but so to give you an idea for heterozygosity, the 2 things that it affected was leaf. Here we're showing leaf length, but the graph shows leaf area, and then tuber biomass, or turion biomass. So we do see an increase in biomass with heterozygosity. It's statistically significant, but when I look at this, I see a heck of a lot of spread in levels of heterozygosity. And the other thing to note is that just in general, is the HWC population tended to be slightly more heterozygous than the SWP population overall, so we have a confounding factor there. And now when we look at leaf area, the reason that said yes, no in the previous graph is that we see an effect in one population, but not in the other. And so there's an interaction between the population from which you came and whether heterozygosity actually affects tuber biomass at all. But again, there's, to me, it's statistically significant, but I look at that spread and I have trouble thinking that heterozygosity is explaining a lot here. So when we look at genotypic diversity, so we're looking at whether an increase in genotypic diversity has an effect on leaf length, number of ramets, tuber biomass, length of flowering, number of flowers in this case. We find that number of genotypes does have an effect on all of these. Genotype identity also has an effect on everything but length of flowering. And then the population source has an effect on everything but leaf length. And for number of ramets, this response changes through time, through the timing of the experiment. So when we planted 8 genotypes together, there were more ramets early on, but then there were no significant differences after that. And that could be that they just crowded out their growing space, and that they would, if they'd had unlimited growing space, they would have continued to put on more ramets, but they hit a limit, and we're not sure. And then for population source, the number of ramets, so the SW Piscataway genotypes produced more ramets until week 12, and then that switched, and at week 24, Hawks Cove actually had more. So we saw early growth in one, and then the other one caught up and actually superseded the other. And so the reason that we're interested in these things like leaf length and ramets is leaf length is going to tell you about the ability to grow up to the surface and basically be in the zone where the light can penetrate even if you have poor water quality. So these might be individuals that are can still photosynthesize even under poor water quality, where number of ramets is going to tell you how able they are to spread out and dominate a site and basically persist through clonal growth. And we do see a trade-off in individuals that either grow long leaves or individuals that are sending out lots of ramets. And so there are some clones do one thing well and the others do the other well.
And longer leaves would be more nutrients? And then one is towards...
Well, one is growing up to the sun, right? Less lateral spread and growing up.
Which allows you to access nutrients better.
No, sunlight. Sunlight.
Right.
Yeah, the nutrients are throughout the water, so.
And then the other one
is spreading out and occupying area.
So that would allow you
Yep.
to access nutrients better.
Nutrients are probably not limiting in this case, so it's basically you're able to just dominate space and come to.
Or maybe sexually reproduce better.
That requires another individual, right? But yeah, so basically, so if you just think of the, if you are you're occupying more space in this season, and then every one of those ramets, every one of those clumps this season is going to produce at least one and probably multiple tubers to then grow up in the next generation, and that's going to, or the next year, not generation, but the next year. And so that's going to give you just more and more space that you can occupy. Where if you're putting your energy into just growing a leaf, you will produce your turions right there, but you're not going to be dominating more and more area. Does that make sense? That's where we're headed. That's what we're interested in, is do you see these having, so most of our experiments have been in the greenhouse, well, our experiments have been in the greenhouse to this point, so we're now interested in how these, so we can just jump to the end of the talk here. I don't need to tell you anymore. So we're interested in how these are, how they show up in populations and what they're doing in the bay to understand whether they're conferring resilience and potential for persistence in different ways in different locations. So. Yeah, so we do see that genotype identity. So certain genotypes do play a large role in the behavior, but also this population source does as well. And it's a little bit confounded, but we do see that certain populations can have a lot of similarities in terms of their behavior. But we've only looked at the relatively few populations, and so we need to scale that up. Okay. So one of the interesting things is the effect on just on flowering, and it's kind of a classic Allelic effect, where if we have only 2 genotypes, we have very few mesocosms that have both sexes. And so in these populations that have few genotypes, it's likely that you're going to get to the place where you can't have sexual reproduction, and then you get into this cycle where, because you can't have sexual reproduction, the clones that are there are going to come to dominate larger and larger areas because there's no option for anything else, right? So, so you have very few mesocosms with both sexes, and then you have female-only mesocosms and male-only, and then no flowers at all. So we have individuals that did not flower during our experimental time. So as we increase to 4 and 8 genotypes, the number of mesocosms with no flowers drops. So the more genotypic diversity you have, the more chances you have that at least one of those genotypes is going to flower. And then the number with both sexes goes up. But we still have this male domination where we have more male flowers than female. But so it's classically effect where you're unlikely to get the opportunity even for sexual reproduction when you have little genotypic diversity. Okay, then our summary of results for the local adaptation part of the experiment. We find that sediment type and population source both have an effect on all of the characteristics that we measured. But to really look at, document local adaptation, you need to have better performance in your home conditions than foreign conditions. And you need to have, the home genotype has to have better performance in its home condition than the foreign genotype. So you have to have 2 conditions met to technically have local adaptation. And we see if we plant the Piscataway plants in their own soil and HWC plants in SWP soil, actually the HWC plants do better in the SWP soil than the SWP plants.
What water are you using? I mean,
it's fresh water. And
I wonder, I mean, you're taking these out of this nutrient bath.
Yes.
This 10 micromolar nitrate.
And they were fertilized. They were fertilized, but the surface fertilized, slow release in the pots. Yeah.
So you tried.
So Osmocote.
Right.
So they were not. These were not nutrient. Should not have been nutrient limited. But they're certainly not bathed like they are probably in the bay.
I just wonder if you're switching your leaf to root ratio in that your sediment, if you're using slow release osmocote with sediments, you're feeding it through the roots, whereas in the bay they're probably getting it surface.
So we top dressed, and so certainly some of that is sinking in through the roots, and some is probably dissolving in the water. But you're probably right. We're not exactly mimicking what's in the bay.
I was wondering if that might be part of it.
Well, the differences I think we're seeing here are, let me finish talking about this and I'll get back to that. So in both SWP plants and HWC did better in HWC soil. So it's not that they switched necessarily their root to shoot ratios, but they both did better in HWC soil, but HWC did better than Piscataway. So what we see is the HWC plants just performed better regardless of the soil they were in, and both populations did better in the HWC soil. So, and this is just much better soil. It's much, there's more fines, it's less rocky, got less shell in it, and so they both seem to do better in this soil than they did here. But the HWC plants did better in the SWP soil than the HWC. So if you're looking for demonstration of local adaptation, we really only see, we meet the definition in here, but we don't meet it for SWP. And so ideally we would have additional populations that we're testing this on, but we weren't able to do that in the first year of our experiment. So does that get back at, and they were all treated the same. So although we were not exactly replicating the bay conditions, which we can't do in the greenhouse, they were at least treated equally. So I think it's more the nature of these two substrates, but that there is a population effect, that the SWP plants are just not as robust as the Hawks Cove. So, okay. So we do have substantial genetic structuring in the bay and its tributaries, and that we have indications that individual genotypes and population sources do affect the ecological performance. And there's some evidence for local adaptation, but not complete. Okay. So one of the next things that's important for restoration is your choice of starting material. And there's, and the reason, one of the concerns people have is whether you stick with local stock or whether you introduce genotypes from other populations. And so there's a big debate in the restoration community to where how you should do your stock selection. And this comes down to whether you think inbreeding depression is more important or outbreeding depression, where you're mixing genotypes from different populations, is more important. And so this work was done primarily by Katja did a lot of the crosses, and then Brittany West did the seed germination trials. So we looked at whether there was evidence for either inbreeding depression or outbreeding depression through crosses between males and females from within the same site, cross sites, but within genetic regions that we found through our structure analysis, and between males and females from different populations and different regions. And so these crosses were done in the greenhouse during the first experiments. And then, so if we look at germination percentages, or the proportion of seeds that germinated, and I will say we also looked at fruit length and seed number, and those did not differ at all. But if we look at germination, if we just look at regions, there's no difference in proportion of germination across regions. And the black is the proportion successfully germinating, and the white is not germinating. If we break that down to populations within region, we see huge differences that are masked if you just look at things at a regional level. And so for our northern Chesapeake Bay here, we range from a small proportion of germination to these three being more similar at about 30%. Some variation, but sassafras is the lowest here. We're in the mid-Chesapeake Bay. We see even more variation across sites, with South Ferry Point having almost no germination. And then again, within the Potomac River, we only had a couple of sites, but we see variation there. And then the York is the York, and it's the only population we had down there, so it's always by itself. So now it's going to get a little bit complicated. So these are within population. So this is crosses of males and females from the same population. If we look at crosses that are from, so these are females within these populations, sired by pollen from either HWC or Piscataway. So this is anything fathered by HWC with a CP mother versus HWC with any of these other mothers. We see no consistent pattern, unfortunately. We see some populations that had higher germination from external pollination sources consistently. And so these are pollinations from within the population versus pollinations from HWC and populations from SWP. So in both cases, the foreign pollen increased germination, which would give you some indication that perhaps there was some benefit of bringing in outside genetic diversity. We see the same thing here in the Horse Landing or the York River population. And remember, this is the one that showed signs of a bottleneck. This was the only population we had surveyed that showed signs of a bottleneck. We also see cases in which the paternal source matters more than within versus among region variation. I should have also pointed out, so HWC is in the central region, and Piscataway is in the Potomac region. So we see effective paternal source. And then we see some effect where there's seems to be a maternal source that's independent of paternal source, where you have, doesn't matter who you get pollinated by, but there's variation based on who the mother is or where the mother came from. So unfortunately with this, what we would like to see is that either you had some signal of, yes, we need to move stuff around and alleviate inbreeding depression, or we have issues with outbreeding depression where you have a decline if we're mixing things, and we see no consistent signal. And when we looked at all the other variables that we could look at to predict, could you say when you might have some sign of needing to do genetic rescue versus not? We couldn't find anything, which is not what you want to tell managers, and that it depends. And so what we were trying to do is look for a way of getting at this question where you wouldn't have to actually go do the genetics, where you could have some guidelines, and we could not find any consistent pattern. So okay. So let me move on. I was just summarizing. So we have differences in seed production and germination success, but few consistent signs. And who the mother and father were seemed to be more important than anything else. It was very specific. Individuals seemed to do well or not. So overall, we have a lot of genetic structuring, and the most indications are that individual genotypes and population sources are affecting ecological performance more than our heterozygosity is. Some evidence, but not strong, for local adaptation, and the effects of crossing varied all over the place. So our view is until we know more, we should be somewhat circumspect about moving stuff all around the bay, and at least stick with the genetic regions that were detected. Okay, so I'm going to close by talking about where we're going next. And so what I'm showing here are, we've zoomed into the North Bay, and the dark patches here are populations of Vallisneria in 1998. And the gray here are lines indicating connections among populations at the 4-kilometer scale. So everything connected by gray, and it looks less solid because there are so many patches, are connected at 4-kilometer scale, which seemed to be, from a lot of the work that we had done, a scale that there was a consistent genetic interaction among these sites. And so, but what we see is if we look at this same pattern over time, and these are years for which Virginia Institute of Marine Science has complete aerial photo coverage, and if we go from 98 through 2000, 2004, 6, 7, so we're progressing this way, what I want you to see here is that we shift a lot in terms of the number of sites and the locations that are occupied, and the connectivity that you would see across those sites. So we have different degrees of isolation and different amounts of area being occupied each year. But if we take the composite of that, if we sum all of the areas that were occupied over that time, we have a much larger area being occupied and much higher degree of connectivity. So what that may indicate is that although in any one year we have a lot of turnover and not all these sites are occupied, that with perhaps slight improvements in water quality, we could have more of those sites occupied over more years, and we would actually have a greater degree of connectivity than would be evident in any one year. So that's where I wanted to end. So, okay.